Professor Salvatori Pece from the European Institute of Oncology in Milano. You were working in stem cells in breast, normal breast and cancerous breast.
Right.
Why?
Because what we believe now that there might be an enticing congruence between normal stem cell and cancer stem cell. So from a conceptual point of view, there is there is increasing evidence that the true responsible cells for tumor growth, for tumor initiation and growth, are stem cell sharing a number of properties with the normal stem cell, for instance, self-renewal ability. So these cells are immortal, but are immortal and quiescent. So they may be the real responsible for tumor growth, but also the real responsible for chemoresistance, given based on their quiescence nature. What we are trying to do is to gain insights into the normal stem cell biology in order to highlight, to provide an outlook on cancer stem cell based on the intriguing hypothesis that the molecular mechanisms regulating stem cell, normal stem cell homeostasis, once deregulated, might be the very same responsible for tumorigenesis. So to make a long story short, from a conceptual point of view, tumorigenesis can be regarded as to normal organogenesis gone awry. So tumors developed like tissue, hierarchically organized according to a morphogenetic program, albeit aberrant. We are trying to understand which molecular mechanisms are underlying the aberrant displaying of the morphogenetic program in tumors.
Any results?
And we have a number of results, and we have identified, we have just obtained the full molecular profile of a normal stem cell, and we have used the normal stemness signature to interrogate data sets, and much to our surprise, using the normal stem cell profile, we are able to subclassify tumors in two different groups, displaying different prognosis, displaying a different degree of differentiation. And in other words, we can now interpret tumorigenesis. Actually, to be correct, now we know that the poorly differentiated tumors are enriched in cells sharing normal stem cell features. I cannot say at the moment that they are cancer stem cell. Operationally speaking, they are. Now we are trying to manipulate those molecular pathways in order to verify that they are the true responsible for the cancer stem cell nature of these cells that we identified. So now we have a molecular profile of normal stem cells that is actually providing us with a number of biomarkers that can be utilized for prognostic and diagnostic and maybe therapeutic purposes in breast cancer. So we can use the molecular profile of stem cell to refine our diagnostic, prognostic and therapeutical potential in breast cancer.
Thank you very much.